It's my pleasure to introduce Dr. Mieke Huijbers and Dr. Sebastiaan Akerboom. So Mieke has a background in molecular biology and biochemistry with a PhD in the field of biochemistry and zymology. In this role, she has obtained profound knowledge and experience in working with enzymes. This included work focused on elucidating the structure, function, and mechanism of enzymes. After her PhD, Mieke conducted postdoctoral studies focused on biocatalysis. In her current role, she is working as a lecturer in molecular biology and biochemistry at Leiden University of Applied Sciences in the Netherlands. Sebastiaan has a PhD in the field of inorganic chemistry and has experience with solid state chemistry and organometallic chemistry. After having worked for 9 years in various research and development roles in corporate, he switched to a position as an innovation chemist, working for an investment firm and focusing on sustainable energy, storage, and generation. And very importantly, Mieke and Sebastiaan are the proud parents of Ivan, who's diagnosed with the UGDH-related disorder. Driven by their personal involvement, they are committed to developing an effective treatment method for this syndrome. And even though I was going to say it's wonderful to have scientists with their expertise also be parents. Of course, none of us want to be in this position at all. And we're all trying to work ourselves out of certain jobs. So thank you so much for the work that you have done today. And Mieke and Sebastiaan, please take it away.
Many thanks. Also, thanks for giving us the opportunity to speak today. I must say, I find many of the stories that we've seen very touching. I recognize a lot of aspects there. It takes me back to our own story. But I also am pleased to see that such an ultra rare disease has such a relatively large community behind it, with parents, scientists, and that they are now all brought together, having the opportunity to interact. So that's great. So yeah, many thanks for also including us and making us part of this whole community. It's an excellent opportunity for us. Now, you already introduced us a bit. I also prepared a slide, but maybe it's for future reference. This is us. Rebecca told us most of what's written here. I think what I want you to understand from this is that we are both parents of a son that I will talk about a bit later, and we have both a scientific background. So that puts us in a certain position when it comes to understanding diseases, but also it gives us a background that forces us to think in solutions and challenge the status quo. But a bit more about that later. I think this is, of course, the main reason we are here. Sorry. It's Ivan. He's a lovely boy. He's very happy, as you can see. He's doing fine, and he's a very cheerful little boy. He likes to play wild physical games, a lot of activity. It really engages him, and I also feel that it helps him to develop. So that's great. He likes to be outdoor, look around. Ride his tricycle. Well, he makes me ride the tricycle, but he has a great pleasure when I do so. So that's him. Yeah, so you have a bit of a face with his name. That was the main purpose of this slide. And I think it's good if I take you a bit, with me on our timeline. When Ivan was born, the whole birth was quite uneventful. Everything went well, so we were happy. Fresh parents, we went home. And nothing seemed really out of the ordinary. Maybe around Christmas. He was born in July. Around Christmas of that year, we started to become a bit concerned. He was not developing according to the plan that you receive when you yeah, when you get a child. There's usually some sort of checklist that you can follow through, and he was keeping up until maybe November. And our concerns grew larger and larger as time passed by. And in January, we were genuinely worried. Like, there could be something wrong with him, but it was still very vague and generic. But with that worry, we still enjoyed our parenthood until March, when we first saw the first spasms. And that prompted us to go to contact the hospital directly. I said, well, this is such a bad sign. We have to take action right away. The first hospital visit in March, they took our worries very seriously, thankfully. And I believe it's even less than a month, but in April of that year, the diagnosis was clear. It was very fast. I recall Dr. Jamuar explained that this is one of the problems, that it takes a long time to get diagnosis. Well, we were actually blessed with a very quick diagnosis, thanks to the whole exome sequencing that was conducted by the hospital. So, then we have our diagnosis. And basically, this slide captures what we were told by our doctors. Your son has a UGDH-related syndrome. It's ultra rare. Very little literature was available. Well, the paper you see an image of here on this slide was actually a very comprehensive overview of what was known at that time regarding this condition. So that was given to us in a hard copy. We studied it completely. And one thing that did not sit very well with us is that Ivan has had a variant of unknown significance. One of the enzyme variants was known, but the other one was, you could question basically whether it was pathogenic or not. And that prompted us to contact Dr. Simpson's group, who was also a co-author of the paper I just showed, in trying to understand basically the biology of the disease. Does Ivan really have UGDH syndrome? Can we be so sure? We challenged our doctor. She tried to reassure us, but she failed kind of. So we needed that extra confirmation from Dr. Simpson's group. And I believe, the results of the work that her group conducted were published earlier this month in this paper. So, now we finally have our confirmation. Yes, Ivan does have UGDH syndrome. Yeah, we kind of knew, but now we are sure, right? That's a part of information that you as a parent would like to see. Now we have clear proof, clear evidence that he is affected by this condition. But then, of course, between now and the timeline I just showed, there's a big gap. We didn't sit still because, don't get me wrong, we were devastated. We were with this diagnosis in hand from happy parents living a relatively worry-free life. We turned into parents that had to deal with all of the stuff that most of the people in this audience are aware about. It changes your life profoundly. And of course, people have coping mechanisms, so do we. And I think it's safe to say that we wanted to do something about it. We wanted to fix it. And of course, trying to understand what is going on is very important. We need that information, we need that confirmation. But we also want to fix the problem, because all that is available at the moment is symptom treatment. You try to suppress epileptic activity in the brain. You give occupational therapy to the child, or maybe supportive treatments for the gastrointestinal tract. I don't know, depending on the extent by which the child is affected, there is symptom treatments. We are convinced that these children deserve more than that, actually. So then we go to the next slide. I think this is showing what happened in between. So in April of 24, as I told you, we had the diagnosis, and we initiated the work on the enzyme research not much later. But our coping mechanism really was trying to fix the problem. So we started to reach out to companies, research institutions, researchers, whoever could help us to build towards a therapeutic approach that can fix the problem to the extent that the child has a fair chance, a better chance in life, a better chance to develop. So lots of emails were sent out. I think in March of 25, we had our first solid contact with an institution that was able to give us really some help, and that gave us a lot of hope, to the point that we started our own foundation, the Moiré Foundation, and that gives us a platform to really start working on the development of a therapeutic approach. Because at some point you can no longer do this type of work as parents, especially when you have to collaborate with different institutions across the globe. You need a more solid platform in the form of a legal entity, and that's our foundation. In case you wonder, the Moiré Foundation is named after the Moiré effect, which refers to the kind of interference pattern that forms whenever the overlaying fine mesh structures create a secondary structure it's a visual effect that you may recognize from television screens. In any case, Ivan seems to like this a lot. We can see it in our mesh light fixtures at home. And also when I play videos of this effect on the TV, he is very much interested. So that's the name. And that's, our mission is written here. I'm not going to repeat it. So we want to develop a curative treatment to improve the quality of life as much as possible. What type of treatment? It was written already in one of the previous slides. We believe in gene therapy, and the reason for that is simple, is a bit of a stretch. You see, we now know the gene where the problem is. There's a small error in that gene, and in fact, we know at the molecular level what is wrong inside the cells of our son. At a very precise level, this is the smallest level that is still biologically relevant. And yet, there is not a thing we can do. We can do only very broad symptom treatment. And I found it very difficult to wrap my head around this, that we know in such great precision what is wrong, and there's nothing we can do, and even our son isn't having a fair chance in life because of this. That's very hard to swallow for me. but at the same time, it also gives you a handle because, yeah, the target for the treatment, of course, is in the gene itself. And yeah, that's why we selected, opted for a gene therapeutic approach. And yeah, Mieke will yeah, tell you a bit more about yeah, our approach and the reasoning behind it. Mieke?
Thanks, Sebastiaan. And indeed, I will take you now, after this introduction of our foundation, along on our, strategy to develop a gene therapy for UGDH syndrome. So given the diversity of our audience, a small introduction in what is actually gene therapy. I think most folks have heard of it, but it's a medical technique that actually treats diseases by correcting the underlying problem in the DNA. So what you can do this in different manners, but what you basically do is deliver a healthy copy of the faulty gene to replace or sometimes fix a missing one. And this is done by using a harmless virus, which can modify. So the virus is not infecting anymore, but it's being used as a carrier to carry the new gene into cells. So in the end, what you do is you fix the underlying genetic course of the disorder instead of just managing the daily symptoms, as just described by Sebastiaan. And the idea is that it's a one-time treatment that can offer long-term effects or lifelong recovery. As has also been mentioned in previous talks, it's always a question on what is the right time range for delivery, how is the natural history of the study, and but we believe that UGDH syndrome is actually a good target for gene therapy. The exact genetic cause is known, and as has been explained also by Lena, it's caused by a single gene and by mutations in a single gene, giving it a direct target. Some of you might have seen this image. It's a figure from the paper we showed before. And what it basically shows is the UGDH gene with all the different variants included in that first research paper. And there are even more known which are not included in this paper. So all UGDH patients have different pathogenic variants, but all on the same gene. So the idea is that with gene therapy, you can target all of these variants with one and the same therapy. So given the neurological manifestations of UGDH syndrome and the function of UGDH in the brain, treatment of the central nervous system is needed to provide a therapeutic effect of a therapy. And as has been described in the previous presentation by Jacinta, the brain is one of the most difficult to reach organs in the human body, or maybe the most difficult to reach organ, which is because of the blood-brain barrier. So this is a barrier which basically separates, the brain from the system of the rest of the body to prevent compounds moving in and out of the brain. But there are many different elegant methods to actually deliver drugs to the brain. Therefore, we are collaborating with Apertura Gene Therapy. They have developed a capsid, a virus, as mentioned before, a carrier for the gene to be delivered into the body. And they deliver the capsid, which specifically can be delivered to the brain. So this capsid, this virus can cross the blood-brain barrier and deliver the gene directly to the brain while being injected through an IV, so by systemic administration. Means no brain surgery needed, and you can just deliver gene therapy through IV, and the gene therapy would be delivered to the brain. You can see this capsid as crossing the brain barrier by using, it basically uses a ferry that is already there. So it uses a so-called receptor on the blood-brain barrier to cross this barrier. And it uses existing biological path to cross this blood-brain barrier. Besides having this advantage of being delivered via systemic administration, it also has the advantage that it spreads. This capsid spreads very well throughout the brain, so it reaches most of all parts of the brain. So what is actually needed from now, from this moment, to move to the development of a gene therapy? Well, we divided the research that is needed into 4 different stages. The first stage would be that we would have to test if in cells, if when you add, so there are cells that have a very low expression of the UGDH gene, and we want to test if we deliver the gene to these cells, do we get expression of the gene, and do we see the UGDH enzyme in the cells? So we basically want to test, can we restore cells? Can we transform cells so that they produce more UGDH? If this works, we can move to the second phase of the research, in which we want to test in mice how much of the gene do you actually need to be delivered to the brain. So we need to determine which dose is needed to deliver, which dose of the gene therapy is needed to be delivered to the brain for an effective therapy. And we also need to check, is there a toxic dose? So at a certain point, you might imagine that if you add too much of a gene, it can have toxic effects. So this is also something we have to establish. So that would be the second phase of the research, testing the dose and safety in mouse models. If this is all tested and found effective and successful, we could move to the large-scale production of the gene therapy, production of the therapy to be used for human trials. And that would move us to the last step, the clinical trying, dosing the first patient or hopefully patients. So this research plan for the preclinical stages and also for the manufacturing is well, almost finished. Our research partners are in place, and we are finalizing these plans. So we are far in developing this research strategy. But each research plan also has a financial side. So we have also been busy setting our fundraising targets. And these fundraising targets are defined by the same stages as mentioned on the previous slide. So we have divided the preclinical stage in which we test in cells, and the preclinical stage in which we test in mouse models, and the manufacturing of the actual drug as fundraising goals. Well, and of course, each step needs to be finished successfully before being able to move to the next step in our research. Well, this is what we have been working on the last, well, year, I would say. So if we wrap this up on our timeline, well, Ivan was born in July 2023, which, well, we defined the stage as the unknown because we had no clue yet what would lie ahead. And as Rebecca said, introducing us, sometimes we are, well, forced or brought into roles which we did not expect ourselves to be, but life throws them at you. So at the end of April 2024, Ivan was diagnosed, and we started directly on getting to know the biology and reaching out to see, is there anything we can do to, well, give Ivan a better chance in life, and hopefully give also other kids with UGDH syndrome a better shot in life. And the past year we have preparing all this, and we are now on, well, on the moment that we are moving to the phase of the research actually starting. So with this month, Ivan's data on Ivan's specific varieties published, we are in a very good place also to start our research. Our plan is almost complete, and we hope to be able to start our research this summer. So we are currently finishing these research plans. We are setting up fundraising efforts, and our website and LinkedIn has gone live, and we are also still fine-tuning our website. It has a Dutch version now, but the English one will follow very shortly. But well, nowadays you can relatively easily translate web pages, although sometimes it gives some interesting translations. Bookmark this website if you want to follow us, and we invite you to follow us on LinkedIn. And if you have any questions, you want to talk to us, or feel free to reach out to our email address listed below, because besides researchers, we are foremost Ivan's parents. We want to help this shiny boy shine even more in his life. So thank you for, again, for the opportunity to speak today. And thank you for listening to us.
Thank you very much, Dr. Huijbers and Dr. Akerboom. It's extremely inspiring to hear your story and to hear your work and to hear how you are taking that devastation and turning it into determination and motivation. We have a question in the chat from Dr. Batani. She asks, which in vivo models are you planning to use?
Okay, thanks. So my name is Andrew Steinzepper. I am the acting chief technology officer for Apertura Gene Therapy. I'm actually just here though today as a volunteer. Can you hear you now? We were able to speak with Mieke and Sebastiaan on that time frame that they showed earlier and help build out that non-clinical development path. The current approach here is to use mice that have the human transferrin receptor knocked in. We've worked with the FDA in the past across a number of programs to get confirmation from the agency that they are a sufficient model for both biodistribution and toxicology outcomes. Since there's no viable disease model for UGDH at this time, the approach would be to use in vitro systems to assess the capability of a payload and demonstrate its potency, and then transition over into safety dose range finding using the human transferrin receptor mice. Like with all of this, there's the opportunity, and I believe the approach, to actually go talk to the FDA and the EMA early and often, because that's actually a free exercise, and it's important that we educate the regulatory bodies about what's going on, and then get their alignment to move this forward. Noting here the work that's happening at Apertura, and with this capsid, we have 3 planned clinical starts in the next 12 months. One for a developmental and epileptic encephalopathy called Frizzle, or FRRS1L, that's slated for the end of this year. Another that was recently announced for MAPT for Alzheimer's and other associated disorders. It would also be at the end of this year. That's from a group called Aviato Bio. Sorry, the first one is from a ultra-rare disease foundation called Finding Hope for Frizzle that we've partnered with. And then a third that would be in the first half of 2027. It's partnered with the Broad Institute of MIT and Harvard for prion disease, which is looking at genetic knockdown. So there'll be multiple programs moving into the clinic, all using that same AAV capsid that Mieke
Finding Hope for Chrissy of Finding Hope for Frizzle is actually tomorrow talking at the end.
Oh, that's awesome. I was just with her last night at a fundraising event. So I'm sure you'll get to hear more about that.
I'm not sure how many of our families how much they've really been able to learn about gene therapy. It is totally exciting. I think it's incredibly promising, but I know there's a lot of risk too that's being worked through. Can you just explain that aspect of things a bit?
So you mean risk in the treatment itself?
Yes.
So one of the risks that one of the big risks is also liver toxicity usually. So one of the advantages of the approach we are taking is using the described capsid, which moves to the brain. So this reduces also liver toxicity. It does move a bit systemically, so meaning it goes to other parts of the body, which is a good thing, I think, because of course the UGDH gene is not functional in any cell of the body. So it does move to systemically, but in lower amounts than the more regularly used vectors. So that's in that sense, we hope to reduce that risk of toxicity. And of course, we have to do good dose testing. And one of the risks that exists is overexpression, which means that if you deliver the gene with a virus to cells, what if you add more of the enzyme than in a healthy individual? What would happen if you have more of it? So that is something we would have to test in the mouse studies to see what happens if you add, more or a bit too much.
Maybe adding on to Mieke's outline here. So all therapies have risks associated with them. One of the things that I usually think about, so my background is developing cell and gene therapies. I've been privileged to be in that position for the entirety of my career. When approaching this normally, we think about liver outcomes, as Mieke outlined. There are also, and this is true for every sort of therapy, there's a class of adverse events called unexpected severe adverse events. So it's never happened before, but we see something. Those are called SUSARs, or sudden unexpected severe adverse reactions. So those occur. I'll note that at this point in time, there are now more than 5,000 children that have been treated with gene therapy for SMA1 or spinal muscular atrophy. I believe Dr. Gringrass actually outlined that earlier during their talk. One of the critical things for reducing risk when using any therapy or therapeutic is reducing the amount that's necessary and reducing the amount of that product that's used. This is one of the things that we spend a lot of time on at Apertura and in the lab of Ben Deverman, where this technology was initially developed. We now are able to use dose levels that are about 50-fold lower than those that are used in the approved products, while still reaching 80% or more of all the neurons in the brain. So with this, we're reducing that dose, allowing for better exposure, and moving things forward. I'll note that there are a number of other products that are currently in clinical trials that use that same receptor, the transferrin receptor, both in pediatric and geriatric populations. TFR1 CAPX would be the first time it's used with an AAV moving ahead, which is really exciting. It's also very scary, and it's important as we do these things to recognize that clinical trials are executed at accredited medical centers with trained and capable staff to make sure that we have a lot of eyes on the ball all at once, because we understand that it's both a critical day for that patient, for that family, but also for all of these communities as we're all waiting and hoping for everyone together for these to move forward. So with that, I'm sure this is also a thing to ask Chrissy about, ask her her thoughts and feedback. If there are other questions, I'm happy to take those offline that are about Apertura. I can also put in the chat, there's some wonderful resources online about cell and gene therapy from ASGCT, the American Society for Gene and Cell Therapy. Dr. Congress, I think, also outlined that. The information from ASGCT is focused on patient advocacy communities and outlining what gene therapy is, how it works, and there are actually some explainers on very specific cell and gene therapies. So obviously there's nothing yet about UGDH on there, but hopefully one day coming soon there will be. There's a question about overexpression and toxicity. So there are a couple of approaches that can be taken. There are actually current gene therapy clinical trials for disorders where overexpression can be toxic. And one of the solutions or attempts to address that is by including additional control circuits in that information that's dropped off that are kind of like limiters. And they're self-imposing, kind of like a flywheel, so that things can't go above a certain speed or amount. The other thing to recognize, and this is true in all cases, there is always going to be such thing as too much of anything. Now, with that and the types of studies that would be executed as Mieke outlined, we'd be starting with mice that have a normal amount of UGDH. So every level that would be tested and assessed will be in addition to that regular amount of UGDH that they have present. So you can imagine they're an extra safety factor that's being added in on top of this when evaluating that. The question for innovation time window. So I remember having this conversation with Mieke and Sebastiaan. And I think the most important thing to recognize is that if anyone tells you that there's an exact time frame, that this is too late or this is too early, the only thing you can be absolutely sure of is they don't know what they're talking about, because we don't have the data on any of this. There are a number of disorders now that are in clinical trials, and actually some of them even about to get approved, where 10 or 15 years ago, we would say there's nothing that you can do for this disease after point X or point Y. And instead, what we've seen is that, one, the brain continues to develop, or other features and structures are continue to mature. And really what ends up happening is we find out what that window is in those clinical trials. For that. I hope that helps answer your question, Dr. Batani. I hope I'm pronouncing your name correctly.
Do you have a sense of like how you could, let's say the first phases are successful, how you would establish eligibility for clinical trial? Like what would those parameters be?
There's some standard things that are always going to be the case for a gene therapy. So one of those is a confirmation of diagnosis. So it's important to make sure that the only people that are included and treated actually have the disease. Thankfully now, though, we live in a day and age where it's possible to get gene sequencing and confirmation. That's a critical one. There are going to be evaluations that are set by the physicians that would be involved with the clinical trial to make sure that the person is healthy enough to sustain any portions of that treatment. There's usually some amount of prophylactic medication that's provided, corticosteroids or other products, to help tamp down the immune system. Because what we're about to do when we treat with the gene therapy is put a lot of virus in, and our body's pretty good and tuned at slapping that down. So usually what will end up occurring is that patients will receive some prophylactic medication. So it's a standard set of confirmation that they don't have they're not allergic to that medication. There's also a portion of like the folks that are involved being able to consent into the trial, the parents being able to consent into the trial and follow through. All of the specifics of inclusion and exclusion would be set along with the regulators that are overseeing the trial and are part of it. A number of clinical trials are online on clinicaltrials.gov that you can follow and see some of those inclusion and exclusion criteria as they exist, and as more trials are coming online, you can look at that. But the official ones will only be listed once the clinical trial is allowed to move forward.
There's one more question, and then I think we'll end for today. So, who asked, what are the potential sites for clinical trial?
This one we have we have not established yet, so we are not able to answer this question yet. No, we are not in that point yet.
What we can say is that if you look at other gene therapies that exist, that have both been in trials and then even approved, so Zolgensma, which is that treatment for SMA1, is now approved in over 40, I want to say it's over 47 different countries across the world. So with this, there is actually a great deal of experience in testing and deploying these sorts of therapies. And to Miike's point, since this would not require complex neurosurgical interventions, it makes it easier and more accessible for clinical execution at hospitals. So it's everything that can happen to make it simpler to move forward.
And this is something, of course, we will look into the coming time frame when our research advances in the later stages.
Yeah, I mean, we have to be honest. Time is of the essence, so we start going, and we will for sure think about this as we move ahead. We have to, we want to, but yeah, getting to the point where we are now took considerable effort, let's say. But for sure, when we have news, whenever we have news, we will keep you updated through our website, through our LinkedIn page.
Well, thank you so much, Mieke and Sebastiaan. Andrew, thank you for supplementing the discussion.